Good day to those that are watching. We are from Team 7, so we will be presenting about the lipids. So first, my name is Angelica, so I will be starting today's presentations by presenting the classifications of lipids. So basically, lipids can be classified into three, which is simple, complex, and derived. Simple lipids are those that are made of fatty acid and alcohol. The most common one we see is triglycerides, which compose of one glycerol and three fatty acid. The next one, we also have waxes, which is the esterifications of long-chain fatty acid with long-chain monohydric alcohols. The next classification we have is a complex lipids, which mainly consists of fatty acid, alcohols, and extra groups. A common one we see, of course, is phospholipids. Examples of those are phosphatidylcholines, phosphatidylethanoamines, and sphingophospholipid. And next, we have glycolipids. The examples of those are cerebroside, which is a ceramide with one sugar residue, and then we have ganglioside, which is a ceramides and alkyliosaccharides and sialic acids. And then the next one we have is lipoproteins. So basically, lipoproteins is a fatty acid and an extra groups without the alcohols. And the examples of lipoproteins are chylomicrons, HDL, LDL, and VLDL. So lastly, we have is derived lipids, which are produced by the hydrolysis of simple or complex lipids. So the first one we have is fatty acid, which can be divided into saturated and unsaturated. And then the next one we have steroids. Steroids come from cholesterols, which are important for the synthesis of sex hormones. And lastly, we have is eicosanoids. They are called as lipid signaling molecules, and they come from arachidonic acids. So examples of eicosanoids is prostaglandins, which are important for inflammations, and thromboxanes, which are important for platelets aggregations. So that is all for the classifications of lipids. Thank you.
Hi, my name is Oin Sing Tian. I'm going to explain the structure of fatty acid and triglycerides. Fatty acid is made up of non-polar long hydrocarbon chain and polar carboxylate group. There are usually even number of carbons and can vary in length. They are mostly hydrophobic due to the long hydrocarbon chain. There are two types of fatty acid, saturated fatty acid and unsaturated fatty acid. Saturated fatty acid have no carbon-carbon double bond, and it is solid at room temperature. For example, palmitic acid. While for the unsaturated fatty acid, they have one or more carbon-carbon double bonds, and it is liquid or solid at room temperature. For cis-unsaturated fatty acid, they are liquid at room temperature, and trans-unsaturated fatty acid, they are solid at room temperature. Unsaturated fatty acid can be further divided into cis and trans. Cis-unsaturated fatty acid have kinks, and that prevent tightly packing of molecules. So cis-unsaturated fatty acids is more liquid at room temperature. For example, oleic acid and linoleic acid. While for the trans-unsaturated fatty acids, they have no kinks, so that they are straighter and more solid at room temperature. For example, elaidic acid and racemic acid. Triglyceride is made up of one glycerol and three fatty acids. They are stored in the body as fat. They are found in adipose tissue, dietary fats, and oils. This is the structure of triglycerides. A triglyceride is formed when one glycerol molecules react with three fatty acids through condensation reactions, forming three ester linkage and release three water molecules.
Unlike simple lipids, which are just esters of fatty acids and alcohols, complex lipids contain additional functional groups such as phosphate group or a carbohydrate moiety. Their primary job is not just for energy storage, but they are also the fundamental building blocks of our cell membranes and the mining sheaths that insulate our nerves. First, let's look at phospholipids, which contains a phosphoric acid residue. Structurally, they are classified based on their backbone. First, we have glycerophospholipids, which uses a glycerol alcohol base, where two fatty acids are attached to the first two carbons and a phosphate group is attached to the third carbon. The hydrogen group is a polar molecule like choline or serine, which is usually attached to the phosphate, creating a highly hydrophilic head and two hydrophobic tails. Next, we have sphingospholipids. For example, sphingomyelin, which uses a sphingosine base, and the structure is a single fatty acid attached to the amino group of sphingosine, forming a ceramide. A phosphate group and a nitrogenous base like choline in sphingomyelin are then attached to the terminal hydroxyl group. Next are glycolipids, which swap the phosphate group for a carbohydrate moiety. These are predominantly glycosphingolipids built on a ceramide backbone. One or more sugar residues like glucose or galactose are linked directly to the ceramide, and you'll find cerebrosides and gangliosides in high concentration in the brain tissue. where cerebrosides contain a single sugar unit, while gangliosides contain complex oligosaccharide chains. For other complex lipid, we have sulfolipids, which typically consist of a sulfonated sugar attached to a diacetyl glycerol backbone. And finally, we have lipoproteins, where it exists as aggregates. The core is a hydrophobic lipids, and the shell is a protective outer layer made of phospholipids, free cholesterol, and specialized proteins called apolipoproteins.
The first function of lipid is as energy storage. Lipids are stored as triglycerides in adipose tissue. They are the most energy-dense macronutrient, providing 9 kilocalories per gram, which is more than twice the energy of carbohydrates. This makes them an ideal long-term energy reserve, especially during fasting or when glucose and ATP levels are low, at which point lipolysis breaks down triglycerides to supply energy. Next, lipids help in structural function. Lipid is essential components of cell membranes, such as phospholipid bilayer, and besides that, cholesterol also help to maintain membrane fluidity and stability at high and low temperature. Lipids also provide insulation and protection. Subcutaneous fat helps maintain body temperature by reducing heat loss. Besides that, it also provides mechanical protection, such as fat around vital organs which act as cushion. And lastly, they also act as electrical insulators, allowing rapid depolarization along myelinated nerves. Lipids are important in the synthesis of hormones and signaling molecules. Some lipids act as precursors of steroid hormones. Others are converted into eicosanoids, such as prostaglandins, thromboxanes, and leukotrienes. Eicosanoids play key roles in inflammation, blood clotting, and immune response. So on the right-hand side, we have omega-3 fatty acids that produce eicosanoids that are less inflammatory or anti-inflammatory. So this in contrast, omega-6 fatty acids, they usually converted into eicosanoids that are pro-inflammatory, which promotes inflammation, platelet aggregation, and stronger immune response. Lipids also help in the transport and absorption of fat-soluble vitamins, which are vitamin A, D, E, and K. So they help to form micelles and chylomicrons that can transport the vitamins.
To the lecturers and my fellow peers, my name is Haskell Yu. My matrix number is A221587. And my topic today is on the lipid digestion. So let's start with an overview. Triacylglycerol, or short form TG, are the major fat in the human diet for digestion. The fate of the triacylglycerols after the digestion on their product, let's say, is to become 2 free fatty acids, FFA, and 1 2-monoacylglycerol, 2 mg per triacylglycerol molecule. The digestion of lipid has 3 sites of enzymatic action, which are the mouth, stomach, and the small intestinal lumen, which is also the main site for lipid digestion. There are also other dietary fat that are digested, which are cholesterol esters, cholesterols, and phospholipids, and we will talk about it later. And we will kick a start with the initial digestion. They involve the lingual lipase, which is produced by the cells at posterior of the tongue. And this is the one responsible for the lipid digestion in the mouth. And the another one is the gastric lipase. It is produced by the chief cells in the stomach. And it is responsible for the digestion of lipid in the stomach also. And both this and lipase, they prefer to hydrolyze short and medium chain of fatty acids, which contains less or equal to 12 carbons. And their digestion are limited due to the low solubility of the substrates, or these triacylglycerols. And after they have completed, go through the stomach, the food will enter into a duodenum of the small intestine. And in this time, the food is known as a chyme. And the dietary fat, it also follows the chyme to enter into the small intestine. And in the small duodenum, the emulsification occurs. And bile salt is the emulsifying agent in this case. It is synthesized by the liver and while stored and released by the gallbladder. There's a common misconception where people may think that bile salt is also produced by the gallbladder, but it's actually not. It's synthesized by the liver instead. And this bile salt, it actually works like a detergent. It binds to the lipid globules and allows intestinal wall muscles to break the lipid globules via the peristaltic movement, which breaks them into smaller particles. This is what emulsification means, meaning the suspension of the smaller particles of the lipid in the aqueous environment of the organ. And this emulsified fat, they have greater surface area available for the enzymatic action. We'll talk about it later. Now, moving on, we have the intestinal digestion. And the pancreas is the main organ responsible for the digestive enzyme secretion into the small intestine. And the main enzyme in this case is the pancreatic lipase. It hydrolyzes fatty acids from the positions 1 and 3 of the glycerol moiety of the triacylglycerol. So the product is the 2 free fatty acids from the position 1 and 3, and also leaving the another one which is remain to the glycerol moiety. So leaving the 2 monoacylglycerol. However, in this case, bile salts, they inhibit the pancreatic lipase by coating the substrate. so that the substrate cannot move into the active site of this pancreatic lipase. And hence, the pancreas also release another protein known as the colipase. It binds to both the dietary fat and the lipase, hence relieving the bile salt inhibitory effect on the lipase, and allows the triacylglycerol to enter into the active site of the pancreatic lipase for the enzymatic action to occur. There's other examples of the pancreatic enzymes for the dietary fat that we mentioned earlier. One is the phospholipase A2. It hydrolyzes the dietary phospholipids in the intestinal lumen. It is initially released as a zymogen, or the proenzyme, and is activated by a protease known as trypsin, which is also released by the pancreas itself. And after it activates through the cleavage to form the actual enzyme, activated enzyme. And the products of the enzymatic action of this enzyme is one lysophospholipid and also one free fatty acid per phospholipid molecule. And the another enzyme is the cholesterol esterase. By the name, you can tell it acts on the cholesterol esters to produce one cholesterol and one free fatty acid per cholesterol ester molecule. Now we have the hormonal regulation of this lipid digestion. Basically, we have two of the hormones. One is a cholecystokinin. It is secreted by the small intestine when the chyme arrives at the duodenum. And it works by signaling the gallbladder to contract to release the bile acids for the emulsification that occurs. And also to signal the pancreas to release the digestive enzymes we mentioned earlier for the lipid digestion in the small intestine. Another one is the secretin. It is released by the small intestine also in response to the acidic chyme entering the duodenum, because we know that the food that before it enters into the duodenum, it passes through the stomach with the gastric juice, which are acidic. So it works by signaling the secretion of the bicarbonate, which is a common buffer found in a biological buffer. It raises the pH of the intestinal lumen to a range of around pH 6. This is an optimal pH for the digestive enzymes we mentioned earlier. And it also neutralizes the acidic chyme by raising the pH. And that's all from me. Thank you.
I'm Logo E. Today I'm going to talk about absorption and transport of lipid. Okay, the first is mouth and esophagus. The digestion of lipid begins in the mouth with the process of mastications and salivations, and lingual lipase start the initial breakdown of lipid, particularly triglycerides. The food is then pushed down into the esophagus, where peristalsis helps transport food to the stomach. In the stomach, gastric lipase is secreted, which continue the breakdown of triglyceride into free fatty acids and diacylglycerol. The acidic environment in the stomach helps in the denaturation of proteins, making the lipid more accessible to the enzymatic actions. In addition, churning actions in the stomach help mix the fats with the digestive enzyme, preparing them for more efficient digestion in the small intestines. After the lipid have been broken down into the free fatty acids, monoglycerides and fat-soluble vitamin A, D, in K, these molecules form aggregates with bile salts called micelles. Micelles are small water-soluble particles that are able to carry hydrophobic molecules like lipid through the aqueous environment of the intestinal lumens. Micelles help transport lipid to the brush border of the enterocytes, intestinal cells, which are located in the small intestines. The micelles bring lipid close to the enterocyte cell membrane, allowing them to pass through the unsterile water layers. It's a thin layer of water on the cell surface and enter the enterocyte via simple diffusions. Once inside the enterocyte, the free fatty acid and monoglyceride are re-esterified into triacylglycerols and cholesterol esters. This process is facilitated by the enzyme acyl-CoA synthetase. After all, they will packing into the lipoprotein. After re-esterification inside the enterocyte, the newly formed triacylglyceride, cholesterol ester, and other lipids are packaged into lipoproteins, specifically chylomicrons. So, chylomicrons enter the lymphatic vessels in the intestinal veins and travel through the lymphatic system before entering the bloodstream at the left subclavian vein. This bypasses the liver initially, allowing the lipid to be delivered directly to the peripheral tissues. So once it's in the bloodstream, chylomicrons are broken down by lipoprotein lipase enzyme located on the endothelial cells of capillaries, particularly in adipose tissues and muscles. VLDL, very low density lipoproteins, are also produced by the liver to transport endogenous lipids. These lipoproteins transport lipid to tissues where they are either used for energy or stored in adipose tissues. That's all from me. Thank you.
Finally, we're going to walk through some of the critical clinical correlations regarding lipid metabolism, such as obesity, familial chylomicronemia syndrome, and dehydrogenases deficiency. So firstly, obesity. This disrupts the lipid metabolism by causing extra fat storage, leading to dyslipidemia. These fats will accumulate in non-adipose tissue, causing inflammation and next insulin resistance and lipotoxicity. Furthermore, familial chylomicronemia syndrome. This is due to the mutation of the lipoprotein lipase, or ApoC2, which will stop lipolysis in the bloodstream. As a result, they will be trapped in the blood and may lead to hypertriglyceridemia. And lastly, dehydrogenases activity, which is the lack of these enzymes, will affect the beta-oxidation of fatty acid to form energy, acetyl-CoA. So this includes the VLCAD, LCAD, and MCAD, and SCAD.